The Viral Vectors and Vaccines Bioprocessing Group, led by Professor Amin Kamen, is part of the Department of Bioengineering at McGill University. Our group is examining how the viral vaccine production process can be accelerated by understanding how cells and viruses interact, as well as the cellular metabolic state needed for maximum productivity. This understanding will help develop the processes and technologies needed to make safe, potent, cost-effective vaccines. The expression platforms are centered around eukaryotic cell-based systems in suspension, namely HEC293, Vero, and insect-derived cell lines. The viruses we are currently working with in biosafety level 2+ laboratories include recombinant adeno-associated virus, AAV, adenovirus, lentivirus, vesicular stomatitis virus, VSV, Newcastle disease virus, NDV, and influenza virus. The lab is equipped with a variety of equipment and resources that enable us to develop novel cell lines and manufacturing processes for the production, purification, and characterization of viral vectors and vaccines. The Applicon bioreactors and control units enable us to operate bioreactors of different capacities, up to 3 liters. These are suitable to generate enough material for small-scale applications and modify conditions for scaling up purposes. The Amber 250 modular is an expandable benchtop system that incorporates up to 8 fully integrated single-use 100 to 250 ml mini bioreactors for process optimization and process characterization. The Nova Bioprofile Flex 2 is a metabolite analyzer which quantifies the metabolites present in the culture, including glutamine, glutamate, glucose, ammonia, lactate, among others. The QX200 droplet digital PCR system provides absolute quantification of target DNA or RNA molecules for EVA-green or pro-based digital PCR applications. The ACTA event purification system is a chromatography system designed for fast and secure development of scalable methods and processes for purification of recombinant proteins and viral particles. The Waters Hplc Alliance system is a reliable and flexible system for high-performance liquid chromatography separations. Its advanced software functionality simplifies method development and reporting for viral quantification and separation of full and empty viral particles.
Hello, my name is Pablo, and I'm a PhD candidate in Professor Amin Kamen's lab. I research viral vectors as tools for the genetic modification of cells. Particularly, I study ways to improve the production of adeno-associated virus, or AAV, that will be used in combination with CRISPR-Cas9 for the generation of cells of immunotherapeutic value. This way, we can generate, for example, CAR T cells, which are T lymphocytes modified in vitro to target and kill cancer cells. Some of the activities I perform during my project include the use of suspension cells to produce viral vectors in shake flasks, tubes, or bioreactors, the purification of these vectors by affinity chromatography using the ACTA system, where the viral particles bind to the column and are eluted using a specific buffer, and the virus quantification by droplet digital PCR, which gives me the number of viral genomes I produced.
Hi, I'm Julia, and I'm a master's student in the lab. I work on the vaccine production part. My project is focused on producing Newcastle disease virus, or NDV, as a viral vector for vaccines, including a vaccine against COVID-19. This virus is typically produced in chicken eggs, so my goal is to establish a production process using cell culture instead. For my project, I work on the upstream part of the process, which means I optimize the parameters to produce the virus, such as conditions for the cell growth and for infection. I produce these viruses in bioreactors at the lab scale, so usually 1 or 3 liters, so that in the future they can be scaled up to industry-scale bioreactors. I also work on analytics, which is developing assays to quantify and characterize the viruses I produce. In summary, our team is building capacity in viral-based treatments and preventions and supporting the translation of discoveries in vaccines and gene therapy. Our work could lead to developing and commercializing vaccines and immunotherapies that will significantly improve Canadians' health. To know more about our team and projects, check our website.